You saw that in that picture of the folding protein. They formed alpha helices, and then the alpha helices folded on themselves to form that nooks and cranny structure I just showed you. Molly. Is that Molly? I got one name down. Okay? That's called the tertiary structure. Now the polypeptide is fully folded, but there is something called a quaternary structure. Number 4, which is not shown here. And number 4 only occurs between two polypeptides. Once they're already folded, can they interact and bind with each other? So if I had another polypeptide here, and it formed interactions with this one that were stable, that would be an example of a quaternary structure. Okay, 1, 2, 3, 4. From simple to more folded, and then to two things binding to one another. That's not truly a folding process at all. The quaternary structure is the ability of two proteins to interact with one another in a stable way. But they're already folded when you do that, most often, not exclusively. But we're not going to talk about the exception. All right, good. So we've already looked at the primary structure. I drew it out for you a bunch of times. I hope you're practicing at home, drawing short polypeptides. Make sure you see those repeating carbonyls and amine groups. Make a note of them. Draw a 10 amino acid structure out and start circling the carbonyls and the amine groups that are found along the backbone of the polypeptide so that you're convinced that they are there and that they're capable of forming hydrogen bonds. Secondary structure now we're looking at. So here's a polypeptide that's starting to fold around on itself, and it's looking for stabilizing interactions, and it's finding them in the alpha helix. So a polypeptide can turn and twist in the proper orientation so that those carbonyl groups and those amine groups that we've been talking about found along the backbone of that polypeptide are close enough to form hydrogen bonds. And the hydrogen bonds are between one amino acid and another amino acid, 4 amino acids down the alpha helix. So it would be, if I numbered amino acids, let's do that, maybe that's better. Number the amino acid. So let's get rid of that. Oops. If I was to label or number the amino acids that can interact this way, I would call this one number 5 and this one number 1. So amino acid 1 can interact with the amino acid 4 amino acids away. And that's because when that helix is twisted into its lowest energy conformation, the carbonyl and the amine groups from those two amino acids are side by side. Now we could look at this structure and try to tease that out if we wanted to. We'd have to determine what that blue ball meant, but we should be able to do that based on the appearance of this R group. So we know the R group is bound to the alpha carbon. So that white thing must be a hydrogen. Has to be, because we know that alpha carbon only binds 2 atoms. One's a hydrogen and the other one's the first atom of a side group, whatever that happens to be. So that must be the alpha carbon. That makes that a nitrogen probably, but it's only got 1 hydrogen on it, so that's a little puzzling because it should have 2 or more. So that's probably a nitrogen. We'll see if that bears out. There's certainly no big oxygen next to it. There's a hydrogen, so it can't be the carbonyl. That makes this gray one a carbon that must have a nitrogen next to it. This then must be the peptide bond here, has to be. That makes that red thing right there the oxygen of this carbonyl bond. So right here's a carbonyl. So that red oxygen is, you can see there's a hydrogen bond between that and this hydrogen, which is attached to a blue ball, which we've identified as nitrogens. So then the only question is, how many amino acids away is it? So if this is amino acid number 1 here,